In this video, we are going to be exploring genetic mutations. 4 types of mutations include missense, nonsense, silent, and read-through. Let's take a look at a DNA sequence and see what happens when these 4 mutations occur. Here's our DNA sequence along with the coded mRNA and amino acids. Something also important to note is the difference between transition mutations and transversion mutations. Transition is when a purine is replaced by a different purine or a pyrimidine is replaced by a different pyrimidine. Transversion is when a purine is replaced by a pyrimidine or vice versa when a pyrimidine is replaced by a purine. Purines are a type of nitrogenous base in DNA and RNA. Adenine and guanine are purines. Likewise, pyrimidines are also types of nitrogenous bases in DNA and RNA. However, these include thymine, cytosine, and uracil. Also, thymine exists as a base in DNA but not RNA. Let's look at a missense mutation example. A missense mutation is a single point mutation in which a single nucleotide changes, and this results in a different coded amino acid. As we can see, the mutation took place in the 4th quadrant with the original codon GCT. By changing guanine to cytosine, this represents a missense mutation as a nucleotide change coded for a different amino acid, alanine to proline. Also, this is transversion as it showed a purine replaced by a pyrimidine. Next, let's see what a nonsense mutation looks like. A nonsense mutation is when a single point mutation turns an amino acid into a stop codon. In the 2nd quadrant, codon TGG changed to TAG. Changing guanine to adenine represents a nonsense mutation as this nucleotide change resulted in the stop codon UAG. In addition, it is transition as a purine was replaced by a different purine. Here's a silent mutation example. A silent mutation is when a single point mutation changes a codon into a synonymous codon that specifies for the same amino acid. In this example, in the 3rd quadrant, AAC mutated into AAT. Changing cytosine to thymine represents a silent mutation as a nucleotide change still codes for the same amino acid, asparine. This example is transition as the pyrimidine cytosine is replaced by another pyrimidine, thymine. Let's look at a 4th mutation, a read-through mutation. A read-through mutation takes place when a single point mutation turns a stop codon into an amino acid. In the 6th quadrant, TGA turns into TCA. This base change, guanine to cytosine, represents a read-through mutation as the nucleotide change turned the stop codon into the amino acid serine. This also allows the sequence to continue coding. Furthermore, this is transversion as the purine guanine was replaced with the pyrimidine cytosine.